I put this slide up here really for two reasons. One is to give a shout out to Greg and Chris for putting up for this most recent distribution map. And then my other reason for putting this map up is to congratulate the state of Iowa. So in 2014, the most recent, the previous distribution map, there was a single county in northeast Iowa that had not been reported as positive for SCN. And I know that all of you were looking eastwards to Illinois, which had already achieved perfection, and you were looking at us with envy and jealousy, and so you finally achieved perfection. So congratulations. So SCN is important. I'll just briefly go through the life cycle. For those of you who don't know, so they start off as an egg. They hatch as second stage juveniles, unlike C. elegans, which hatches as a first stage juvenile. Those J2s will find a root, they will infect, they'll go into the root, they'll set up a feeding site, and once they set up that feeding site, they don't move. So they'll start feeding, and they'll molt into a J3, they'll continue to feed, and then they'll molt into a J4. Now during the J4, the males will actually start to differentiate into sort of they'll differentiate back into a vermiform stage, whereas the females will grow and grow in size, okay? So during this development, both males and females are found in the root. They both grow in size. However, during that last developmental stage, the females will continue to grow in size and become sort of these pearly white structures that you can see on the outside of the root, whereas the males will return to a vermiform stage during that last J4 stage. This is actually a J4 that is starting to break through the old this is an adult male that's breaking through the J4 cuticle. So there were a couple of questions that I'd been interested in and from the beginning looking at SCN. So one is this sort of increase in size. Why do they get bigger in width? So most nematodes don't do this. So even Ascaris, which is really long, it's 1.5 feet length, it grows proportionally in size. Okay, it grows in width as well as length. But SCN grows more in width. Why does it happen? There's a couple of plant parasitic nematodes that do this. The other question is, why does SCN become sedentary? And I'm gonna talk about that primarily. Hopefully, if I have a little time, I'll talk about the fat size of SCN. So why does SCN become sedentary? So I mentioned they'll set up this feeding site, they'll start to feed, and then they don't move. The females will never move again. They'll stay attached to that feeding site. Okay? They remain sedentary forever. The males, however, will resume mobility after that last molt, and they'll go in search of females. So occasionally in the literature or in talks, you'll hear people talk about this phenomenon when they're introducing SCN, and they'll say that SCN undergoes muscle atrophy or muscle degeneration. Now, if you look in the literature, there's no data that actually supports that. So 40 years ago, a guy named Alan Bird, a nematologist down in Australia working with root knot nematode, which also has a similar developmental pattern, he did some nice behavioral work where he dissected out root knot after it had been infected and tried to see if it would be mobile, and found that, sure enough, it's not going to move. Once it's established that feeding site, he could take it out of the root, and it doesn't move. It can't reinfect. Okay? And similar unpublished data has been shown with SCN. About 30 years ago, Alan Bird had a book chapter where he talked about his earlier results, and he suggested that this could be due to muscle atrophy. And from that suggestion, people have then said, well, it's probably due to muscle atrophy. But nobody had ever looked, and so I said, well, let's try and look and see what's actually happening. So for about the last year and a half, we've been looking at the neuromuscular system of SCN. Some of this had been known based on work by a nematologist at the USDA, especially at the J2 stage, but then some of these later developmental stages, we have some new data to present. So the first thing we did was light microscopy. So we used a fluorescent dye called phalloidin. This binds to filamentous actin, which is highly enriched in muscle tissue. Okay, so we would fix animals. These are second stage juveniles. Fix them, expose them to the phalloidin, hit it with our light, and we see these nice, well-defined muscle structures. This is similar to the muscle in C. elegans. Okay, so everything looks nice and intact and pretty beautiful. So then what we did was we took Alan Bird's tactic, which is to let these juveniles, not these particular ones, but let second stage juveniles infect the roots for a certain period of time, allow them to set up their feeding site, and then carefully dissect open the roots, and then let's look at the muscle tissue again. So one of the interesting things about this phenomenon is that, okay, they're not moving, but they're still feeding. So in order to feed, they need to have some muscle, right? Okay, so we first looked at the esophageal muscle, and what we found was that was perfectly intact. So there are muscles within this bulb here called metacorpus, which are beautiful, intact. This is late J2, early J3 stage after the feeding site has been established. You can see it's starting to gain dimension and width. And then the muscles around the stylet are perfectly intact. Everything looks beautiful. When we look later in development in females, so this is early J4 female, we can focus in. If I tried to show you the full overlay with all the GFP channel as well as our GAPI channel, the GFP would be so faint. We see these tiny, tiny faint lines, which we believe are still muscle tissue that are there, but have definitely undergone degeneration. So the width of this is approximately 1/10 the width that it is in the J2 stage. So it does appear that there's atrophy that's occurring in the female. These are just muscle nuclei, so the muscles do not completely degenerate. It doesn't appear that there's any apoptosis undergoing in the muscle tissue during this, at least by J4. So I mentioned the males become immobile, but then they regain their mobility later on. So what happens to the muscles in males? Well, they show an increase in disorganization. However, the muscle tissue does not seem to atrophy as much as you would see in the females. So we can look, this is a J4 male, and we see on one side, these are two quadrants. You would see, basically, if we focus through the animal, we would see another two stripes of muscle tissue. We could focus in. We don't see these nice diamond-shaped cells. It seems to be a more continuous line, and the muscle filament doesn't seem to be as nicely organized, but it does seem to be more intact than in the female. So it seems that there is some sex-specific muscle degeneration that's going on. So this was all really nice, but I was kind of worried about using a light microscopy marker, in part because getting the stain into the animals is not always foolproof. Sometimes we would do the experiment and we wouldn't see any fluorescence in there, and so we really wanted to get sort of secondary confirmation of this. And so what we've really been working on recently is EM data. This is just neuronal structures. There is a decrease in the number of ventral nerve cord nuclei, although you still see the ventral nerve cord there. So we've been doing some TEM process, transmission electron microscopy. So this is similar to what they did in C. elegans. You fix the animal basically in a plastic block, and then you section it into these 70 nanometer sections, put it under a TEM, and you should see slices like this, something like this. And we're using sort of it's not that new, but a relatively new fixation method of high pressure freezing, which gives you a little bit better quality of after the fixation process. So we started off with our second stage juvenile animals. So when we look at around the ventral nerve cord, so basically what I've done, I've taken our cartoon here, and then I've taken our EM micrographs and pseudo color overlaid them with the same color. So the purple